Welcome back to the deep dive. Today we're doing something a little bit different than our usual fare. Normally we look at a specific trend, a historical event, or maybe, a piece of breakthrough technology.
Right.
But today we are looking inward. We're looking at what you might call the engine room of you.
It really is the black box inside the black box, if you want to look at it that way.
Exactly. We're talking about your values.
Mm-hmm.
We're talking about that gut feeling that tells you to trust a stranger,
Hmm.
or the sudden flash of a memory from 10 years ago that changes your mood instantly.
Yeah, how you decide what actually matters in a world that is just constantly screaming for your attention.
Right? We're looking at an absolute enigma of neuroscience.
It is an enigma. It's a region that has baffled neuroscientists for decades, really, because it doesn't just do one thing. It seems to be involved in everything that makes us human, from how we pack a suitcase to how we treat our neighbors.
We're talking, of course, about the ventromedial prefrontal cortex,
Hmm.
the VMPFC.
The VMPFC, yes.
And I have to say, looking at the massive stack of research we pulled for today, ranging from the early 2000s all the way up to papers published in late 2024, this isn't just about anatomy. This feels almost philosophical.
It borders on the philosophical, absolutely, but our mission for this deep dive is to ground that philosophy in hard biology.
Right.
We are going to move beyond the simplified kind of high school textbook view that the frontal lobe is just for thinking or doing math.
Yeah, the logical brain versus the emotional brain myth.
Exactly, that old dichotomy. We are going to dissect the architecture, the connectivity, and the function of the VMPFC. We need to understand why this specific chunk of tissue is what I like to call the conductor of context.
A conductor of context. I like that a lot. And to hook you, the listener, right away, let me pose a few questions that popped up while I was reading through this material. First, why do you decide to help a stranger, even when it costs you significant physical effort?
That's a big one.
Second, how do you know that an apple sitting in a kitchen is a snack, but an apple in a still life painting is art?
And the third one, which is the real paradox here.
Right. Why does this specific, absolutely crucial part of the brain essentially turn off when you focus hard on a math problem?
Those are the exact right questions to frame this. And the answers lie in some pretty complex concepts we're going to unpack today, things like hidden state representations and the affective connectome.
The affective connectome.
Right, because it turns out the VMPFC is the ultimate link between your memory and your emotions.
Okay, let's unpack this. But before we get into the heavy functionality, the software, if you will, we have to figure out the hardware, where this thing actually is.
The geography.
Yeah, because reading the review by Suzanne Haber and colleagues, this is from the journal Neuropsychopharmacology, published in 2021. The paper is called Prefrontal Connectomics, From Anatomy to Human Imaging. It sounds like the VMPFC is suffering from a bit of an identity crisis.
It is, quite literally. Suzanne Haber, who is really a titan in the field of connectomics, highlights a major issue in the literature.
Which is what?
That VMPFC is often used as a functional label rather than a strict anatomical one.
So people just point at a scan and say, there it is.
Exactly. If you look at an fMRI brain scan, researchers often point to a glowing blob of activity behind the eyes and say, that's the VMPFC.
Yeah.
But anatomically, the boundaries are much messier.
So it's not just one clearly defined organ with sharp borders, like the heart or the liver.
No, not at all. It's a coalition of regions. Haber defines it by looking at the medial wall, which is the flat part of the brain between the two hemispheres, and the ventral or bottom surface.
Right behind the forehead and above the eyes.
Yes. And specifically, we are looking at Brodmann areas 14, 25, and 32.
Okay, hold on. Brodmann areas. For those who aren't neuroanatomists, give us the quick gloss on what that means.
Sure. Corbini and Brodmann mapped the brain way back in the early 1900s.
Based on what?
Based on cytoarchitecture. Basically, he looked at slices of the brain under a microscope and categorized regions based on the cellular structure and organization.
So different cell types mean different areas.
Broadly, yes. So area 25 is the subgenual area. It's deep, low down on that medial wall, and heavily linked to mood regulation.
Okay.
Area 32 is the pregenual area, which sits slightly higher up, wrapping around the corpus callosum. And area 14 is right on that medial strip facing inward.
But the Haber paper mentions a conflict with the neighbors, right?
Correct. The label VMPFC also often grabs parts of area 10, which is the very front pole of the brain, and areas 11 and 13, which sit on the orbital surface.
The orbital surface being right above your eye sockets.
Yes, the roof of the orbits. This is where the conflict comes in. Some researchers lump the orbitofrontal cortex, or OFC, in with the VMPFC as one big regulatory block.
But others separate them.
Right. Others keep them strictly separate because their connectivity profiles are actually quite distinct.
For the sake of our deep dive today, based on the Haber review, we are focusing on that medial wall and its ventral extension.
Just those specific structures.
But why does this specific geography matter so much? Why do we care if it's area 14 or area 32?
Because in the brain, location dictates connection. This region is literally sandwiched between the deeply evolved emotional systems in the subcortex, like the amygdala, and the higher-order cognitive systems on the outer cortex.
It's the physical bridge.
Exactly. And in 2024, Ben Shalom published a really fascinating paper in the Neuroscientist. It's titled, "Four Streams Within the Prefrontal Cortex Integrate." He proposes the four streams model.
I saw that one. It sounded like a new way to visualize how information flows into the front of the brain. Instead of just static spots lighting up, he talks about continuous streams.
Which is a much more dynamic way to look at it. Ben Shalom proposes that the rostral PFC, so those parts of area 10 and 11 we just mentioned, handles elementary computations.
Elementary meaning simple.
Foundational might be a better word. This is distinct from the heavy lifting of working memory that happens up in the dorsolateral prefrontal cortex.
The dorsolateral being higher up and on the sides.
Yes. So Ben Shalom uses this stream analogy. Imagine information flowing from the temporal lobe, which is the part of your brain that tells you what objects are.
The semantic dictionary.
Right. Information flows forward from the temporal lobe into these ventral streams.
So the temporal lobe says, hey, that is a coffee cup, and sends that info forward to the VMPFC.
Yes, but the VMPFC doesn't just passively see the cup. It's catching this stream of sensory information about objects and memories, and it's tagging them.
Tagging them with what?
With value and context. It does this before the information even reaches the level of complex, conscious decision-making.
So it's the first filter of, does this matter to me right now?
Precisely. It attaches personal, emotional relevance to the raw data of the world.
That distinction is incredibly helpful. It's not doing the complex math to figure out how to grab the cup. It's deciding if the cup is worth grabbing in the first place.
That's a perfect summary.
But this brings us to that paradox I mentioned in the intro. If this area is so important for deciding what matters and processing context, why does it shut down when we start doing a demanding task?
Yes. This is one of the most famous and initially confusing findings in modern neuroscience.
Walk us through it.
It goes back to a seminal paper by Dozenbach and colleagues from 2006, published in the journal Neuron. It was called A Core System for the Implementation of Task Sets.
2006, so early days of fMRI network analysis.
Very early. They were looking at task-based fMRI. They found that when you ask someone to do something cognitively hard, like a complex logic puzzle or a working memory test,
The VMPFC goes dark.
it significantly deactivates. The bold signal, the blood oxygen level dependent signal they measure in fMRI, drops below the baseline resting state.
Relative to baseline, yes. So while the task-positive networks, the areas doing the heavy lifting for the puzzle, light up, the VMPFC essentially turns off.
It gets actively suppressed.
So it literally works best when we aren't working.
It supports what we call the default mode. When you aren't focused on a specific external goal-directed task, your brain doesn't just turn off, it defaults to this internal network.
The default mode network, the DMN.
Exactly. But the 2024 update from Dozenbach and Rachel in the journal Nature, nearly 20 years after that original paper, really refines this concept. The paper is called the Brain's Singulo-Opercular Action Mode Network.
Okay, let's break that 2024 paper down. Action mode versus default mode. How has the thinking evolved?
Well, we now clearly see the VMPFC as the anchor of the default mode network. This network is heavily engaged in self-referential processing.
Meaning thinking about yourself.
Right, thinking about you, your history, your current emotional state, your plans for tomorrow. It's recollecting prior experiences.
So basically daydreaming.
Constructive daydreaming, mental simulation, building a narrative of self. But here is the critical physiological part from the 2024 paper.
Lay it on us.
To engage in goal-directed behavior, to enter what they call action mode. The brain must attenuate, it must turn down the VMPFC.
It has to get out of its own way.
Exactly. If you don't turn it down, you get interference.
Interference, like, I'm trying to read this report, but I keep getting distracted by my own thoughts.
That is precisely it. If you're trying to solve a math problem, but your VMPFC is still firing away, replaying that embarrassing thing you said at dinner last night, you simply cannot focus on the math.
So the rumination overrides the cognitive processing.
The Dosenbach and Rietschel paper argues that failure to attenuate the VMPFC directly leads to performance errors. You make mistakes because you are internally distracted.
So mechanically, who flips the switch? If I need to stop thinking about dinner and start working on the spreadsheet, what tells the VMPFC to shut up?
That's where the salience network comes in?
The salience networks.
Yes. There's a brilliant review by Venad Menon in Neuron from 2023 titled 20 Years of the Default Mode Network, a Review and Synthesis. He details this dynamic switching mechanism perfectly.
Okay, how does the switch work?
The salience network is anchored primarily in the anterior insula and the anterior cingulate cortex. You can think of it as the brain's ultimate toggle switch.
It detects what's salient, what's important.
Exactly. It detects that an external task suddenly requires your full attention. When it detects that, it sends inhibitory signals to suppress the VMPFC so the executive control networks can take over.
It actively silences the internal monologue so you can deal with the outside world.
Yes, it hands the microphone over to the dorsolateral areas.
And what happens if that switch is broken?
Then you get stuck.
Stuck in the default mode.
Yes. Menon points out that dysfunction in this specific neural switch is heavily implicated in major depression and anxiety disorders.
That makes an incredible amount of sense.
In depression, the VMPFC often doesn't deactivate properly when a person tries to engage in a task. It stays hyperactive.
So you get stuck in that self-referential loop, just endlessly ruminating on negative thoughts.
Yeah.
And you physically cannot switch into action mode to engage with the world.
Precisely. The VMPFC is functionally stuck on when it should be off. You are trapped in your own context.
It's not just a psychological feeling of being sad. It's that your biological mechanism for stopping the focus on yourself is physically jammed.
The network dynamics are failing to coordinate.
This transitions perfectly into what the VMPFC is actually computing when it is supposedly on. Because for a long time, I thought the VMPFC was just the brain's price tag.
A price tagger.
Yeah, like an economic value center. I see a donut, my VMPFC says value high. I see a plain salad, it says value low.
That is what we call the old view. It's the economic value theory of the prefrontal cortex.
And is it wrong?
It's not wrong per se. It definitely does encode value, but it's just radically incomplete.
So what's the new view?
The new view is beautifully detailed in a 2024 review by Moneta and colleagues in Trends in Neurosciences. The paper is Representational Spaces in Orbitofrontal and Ventromedial Prefrontal Cortex Task States, Values, and Beyond.
Okay, task states.
Right. They argue that purely computing value is too simple. They propose that the VMPFC is actually encoding task states.
Task states. This sounds a bit like computer programming terminology.
It is very computational, drawn from reinforcement learning models. Think about the hidden state concept.
What makes a state hidden?
In the real world, the rules of a situation are rarely obvious or explicitly stated. We have to constantly infer the situation, the state, based on subtle contextual clues.
Give me an example of a hidden state.
Moneta uses a great one in the paper. Imagine an apple.
Okay, I am picturing a standard red apple.
Now imagine that exact same apple sitting on a plate in your kitchen. What is the state of that environment?
The state is snack time, eating, a healthy food choice.
Right. Now imagine that same red apple tucked inside a suitcase full of folded clothes.
The state is packing for a trip, or maybe smuggling fruit across an agricultural border checkpoint. But it is definitely not eating right now.
Exactly. The object itself, the sensory visual input of the apple on your retina, has not changed one bit. But the state representation is completely different.
Because the context changed everything.
Moneta and colleagues argue that the VMPFC is responsible for actively holding this state space in your mind.
So it integrates the stimulus, which is the apple.
And the context, which is the suitcase.
And your memory, knowing that apples get crushed in luggage.
Yes, it binds all of this together to define your current reality.
So it's not just assigning a static numerical value to the apple, it's assigning a dynamic value to the entire apple and suitcase situation.
Yes, and this is where the cellular biology gets really interesting. Moneta discusses a phenomenon at the neuronal level called mixed selectivity.
Mixed selectivity. What does that actually mean for a single neuron?
It means individual neurons in the VMPFC don't just have one job. They don't just fire exclusively for apple or exclusively for good.
They don't have a single tuning curve.
Right. They fire for highly specific, complex combinations of features. You might have a specific neural population that only fires vigorously for the state of apple in suitcase while hurrying to the airport.
That is incredibly hyperspecific.
It has to be. This complex, high-dimensional representation is what allows for flexible, intelligent decision-making in humans.
Because if you just had a simple rule where apple equals good, you would try to eat the apple out of your packed suitcase in the middle of a rush.
Exactly. We don't just blindly pursue rewards. We pursue rewards that are appropriate to the current invisible context. The VMPFC is the internal architect of that invisible context.
That is a massive conceptual upgrade from just being a simple price tagger. It's essentially running a continuous multivariable simulation of what is happening right now based on 1,000 different sensory and memory clues.
And that simulation includes the most unpredictable variable of all.
Other people.
Yes. This brings us straight into the social brain. Because if the VMPFC is building a predictive model of the world, a huge percentage of that world is social interaction.
Yeah.
Other people are the ultimate hidden state.
You never truly know what's in someone else's head. You have to infer it.
Constantly.
This brings us to a really standout paper from 2024 by Lockwood and colleagues, published in Nature, Human Behavior. The title is Human Ventromedial Prefrontal Cortex is Necessary for Prosocial Motivation. This was a study on patients with brain lesions, right?
Yes, and lesion studies like this are rare and incredibly valuable. It is very hard to find patients with focal localized damage just to the VMPFC without widespread damage elsewhere.
How many patients did they find?
They gathered a cohort of 25 patients with focal VMPFC lesions and compared their behavior to healthy controls. They specifically wanted to test pro-social motivation.
How do you rigorously test that in a lab setting? Do you just give them a questionnaire and ask if they like donating to charity?
No. Self-report questionnaires are notoriously unreliable for social behavior. People lie to look good.
True.
They used a behavioral task involving real physical effort. Participants had to choose between a rest option, which offered a very low monetary reward, and a work option.
What was the work?
The work meant squeezing a handheld dynamometer, which is a force grip device that measures physical exertion.
I've seen those. They get exhausting quickly.
They do. Squeezing it hard provided a high monetary reward.
Okay, so work harder physically, get more money. Standard effort reward paradigm.
But here is the brilliant catch in their design. Sometimes the reward money was going to themselves. And sometimes the reward money was going to another person, a completely anonymous stranger.
So they literally had to squeeze a metal bar, exerting physical pain, to earn money for someone they didn't even know.
Exactly. It cleanly measures the physical cost you are willing to pay just to be nice.
And what were the results?
The results were stark. Lockwood's team found that patients with VMPFC damage were significantly less prosocial across the board.
Meaning what exactly?
They earned less money for others, and, crucially, they exerted significantly less physical force on the dynamometer to help others compared to the healthy control group.
So if you lose your VMPFC, you just become inherently selfish.
As a general broad strokes conclusion, yes. But, and this is a massive but, Lockwood's team did something really sophisticated with the imaging.
What did they do?
They mapped the exact locations of the lesions to specific subregions within the VMPFC coalition we talked about earlier.
Oh, breaking it down by Brodmann areas again.
Yes, and they found a behavioral dissociation that absolutely blew my mind when I read it.
What stands out to you about this dissociation?
Well, damage to the medial area, specifically area 14 right on the midline, led to the decreased prosociality we just discussed. They acted more selfishly.
Okay, that fits the overall finding.
But damage to the lateral part of the VMPFC region, specifically area 13 out on the orbital gyrus, actually resulted in increased prosociality relative to the other patients.
Wait, let me make sure I'm hearing this right. If you damage the side of the VMPFC, you become more generous.
You become paradoxically hyper-prosocial.
That is wild.
It suggests the VMPFC isn't a monolith with one single goal. It's a push-pull regulatory system.
A balancing act.
Right. The medial part seems to drive the intrinsic helping behavior, the biological do-good signal, while the lateral part might be constantly calculating the cost to yourself, the self-interest factor.
So if you physically knock out the self-interest calculator in area 13 with a lesion, The medial part just runs unchecked, and you have everyone regardless of how exhausted it makes you.
That's the leading hypothesis. The internal referee that normally balances help others versus conserve energy for myself is broken.
That is fascinating. And Lockwood also found something critical about effort in general, didn't they?
They did. The patients with VMPFC damage showed a profoundly reduced sensitivity to effort overall.
Meaning they just didn't want to work.
They were far less willing to work even when the physical effort required by the task was extremely low.
So the VMPFC is a motivation engine too, not just a context engine.
Specifically, it energizes action. It takes the context and calculates if the required effort is worth it to change that context.
It provides the activation energy.
If the VMPFC is damaged, the brain seems to default to a state of not worth the trouble, especially when that trouble is for someone else's benefit.
It effectively removes the oomph behind our good intentions. We might know we should help, but the body won't move.
Oof is a non-technical term, but clinically very accurate. It provides the motivational spark for social behavior.
Okay, so we've got the anatomy, we've got the default mode paradox, we've got hidden context states, and we've got social motivation. Now I want to look at the wiring diagram.
The connectome.
Yes, how does this one region actually talk to the rest of the brain to pull all of this off simultaneously?
For this, we look at a 2023 paper by Edmund Rolls and colleagues in the journal Cerebral Cortex. The paper is called The Human Orbitofrontal Cortex VMPFC and Anterior Cingulate Cortex Effective Connectome.
They used a specific type of analysis, right? Effective connectivity on a large sample.
Yes, 172 humans.
Quickly, what is the difference between effective connectivity and regular functional connectivity?
Good catch. It's a vital difference. Most fMRI studies you read about look at functional connectivity, which just means area A lights up at the exact same time area B lights up.
It's just a correlation. They are dancing to the same song, but we don't know who is leading.
Exactly. Effective connectivity, using dynamic causal modeling, measures the actual direction of influence.
Oh, so it proves causality.
Right. It says area A causes area B to light up. It gives us the actual direction of information traffic on the neural highways.
Okay, so looking at that traffic map, where is the VMPFC sending its strongest signals?
The strongest directed connectivity originating from the VMPFC, specifically from areas 10V and 9-meter, is directed firmly toward the hippocampus and the entorhinal cortex.
The major memory centers deep in the temporal lobe.
Yes, this is a crucial functional pathway. This provides a direct, causal route for reward-related information to enter the memory system.
The VMPFC is basically telling the hippocampus, hey, this moment matters, record this.
Exactly. It's the highlighter pen for our episodic memories.
And specifically, it helps us remember where good things happened, right?
Yes, it links value computation to spatial navigation and episodic memory. For example, if you stumble upon an amazing bakery on a side street, your VMPFC ensures your hippocampus logs those specific spatial coordinates with a massive positive value tag.
So you can find your way back to the croissants tomorrow.
Exactly. It guides future behavior based on past rewards.
And where is the VMPFC getting its incoming information from before it talks to the memory centers?
Both the Rolls paper and the Haber review highlight massive input streaming in from the temporal pole and the auditory association cortex.
The temporal pole handles semantic memory, right? Knowing what objects and concepts are?
Yes. So think about the grand division of labor in the frontal lobe. The dorsal prefrontal cortex, the top part, is heavily connected to the motor and premotor areas.
It's obsessed with how to do things, the mechanics of action.
Right. But the ventral part, the VMPFC, is intimately connected to the memory and semantic systems. It informs what we are doing and, crucially, why we are doing it.
That is a really elegant distinction. Dorsal's the how, ventral is the why.
It's a bit simplified, but functionally it holds up very well. The VMPFC integrates the what from the temporal lobe, the where from the hippocampus, and the pass rules from memory to output the final value.
You can't talk about the brain doing all this work without talking about the fuel, the chemical messengers.
Neurochemistry.
Right. What is the neurochemistry actually running this conductor of context?
A 2024 review by Boyle and colleagues in the journal Brain Sciences breaks this down beautifully. The paper is monoaminergic modulation of learning and cognitive function in the prefrontal cortex.
Monoaminergic. So we are looking mainly at the big ones, dopamine and serotonin.
The usual suspects.
But they do different things in different brain areas. Be specific. What are they doing here in the VMPFC?
Their roles here are highly specific. Let's start with dopamine, specifically acting on D1 versus D2 receptors. Dopamine heavily modulates.
So clarity of thought.
Exactly. High signal to noise means the important context, the true hidden state, stands out sharply against the background noise of random neural firing.
So if your dopamine is dysregulated in this specific area, the context gets fuzzy.
Yes, you might completely miss a subtle social cue or misinterpret the hidden state we talked about earlier.
You might actually try to eat the apple out of the suitcase.
Or more likely, you might fail to realize that your friend's sudden quietness means they are deeply upset with you. You miss the contextual clues that define the social state.
And what about serotonin?
The Boyle paper highlights serotonin, or 5-HT, as absolutely critical for impulse control and processing punishment within the VMPFC circuitry.
Processing punishment. How so?
Remember how we said the VMPFC helps us constantly update our models and learn from mistakes?
Yes, updating the state representation.
Serotonin seems to carry the heavy signal when things go wrong. It helps the VMPFC update its representation to say, okay, touching that hot stove was a terrible idea. Let's heavily devalue that action so we never do it again.
So we are back to clinical relevance again. This chemical balance is exactly what psychiatric drugs are trying to target to fix the network switch issues we discussed with the default mode network.
Right. If serotonin and dopamine aren't perfectly balancing the circuit, the salience network can't cleanly toggle the VMPFC off.
You get the endless rumination of depression or the paralyzed panic decision-making of anxiety.
Because you are chemically unable to update your context. The software is caught in a loop because the chemical hardware is failing.
Man. So let's try to synthesize all of this incredible research. We started this deep dive by calling the VMPFC the conductor of context.
We did.
After diving into Haber's anatomy, Ben Shalom streams, Dozenbach and Menon's network switches, Monetta's hidden states, Lockwood's social effort, Roll's connectome, and Boyle's chemistry. How does that title hold up?
I think it's more accurate than ever, honestly. Think about the sheer flow of data. The VMPFC takes raw sensory inputs, the objects from the temporal lobe, the identities of people from the social processing areas.
It gathers all the pieces on the board.
It consults the vast library of the hippocampus to see what these exact pieces meant in the past. It checks the internal state of your own body via the insula. Am I hungry? Am I exhausted?
And
then it uses that incredible mixed selectivity of its neurons to create a completely unique, bespoke representation of the current moment.
the hidden state.
Yes, and based strictly on that hidden state, it decides the ultimate value of an action.
And it decides if we should get off the couch, burn our own energy, and help a stranger.
Yes. It is the part of the brain that functionally makes us good in the pro-social sense and motivated in the physical effort sense. It bridges the gap.
It connects the cold, hard facts of the sensory world with the warm biological drive to actually do something about them.
It's the bridge between, I see a person falling, and I need to reach out and catch them.
And it somehow does all of this while seamlessly managing its own activity levels, automatically shutting down when we need to focus on a spreadsheet and lighting back up when we have a quiet moment to reflect on who we are.
It is an absolute marvel of evolution.
Here is where it gets really interesting for me, and I want to leave you, the listener, with a bit of a provocation based on all this.
Okay.
We talked heavily about Lockwood's finding that VMPFC damage makes us less pro-social because we can't properly value the effort required to help.
Right.
And we talk about Monetta's concept of hidden states, how we need rich context to know how to act.
Where are you going with this?
Well, look at our modern life. Look at what you are probably doing right now. We spend hours and hours a day in a purely digital environment. Social media feeds, Zoom calls, endless text messages.
A very low context environment.
Exactly. These environments aggressively strip away physical context. They strip away all the subtle apple in the suitcase clues. On a screen, we don't see the micro expressions of body language. We don't smell the environment. We don't share the physical space.
That is a genuinely worrying thought.
If the VMPFC is biologically responsible for representing hidden states and generating pro-social motivation based on that rich context, could our modern digital environment be functionally leaning our VMPFC?
That is a profound and terrifying question.
Right, because if the input to the VMPFC is continually degraded, if the context is just flat pixels on a glass screen, then the state representation it builds is going to be incredibly incomplete.
We might entirely fail to trigger that prosocial response because the VMPFC simply doesn't have enough high-quality data to build the empathy state.
We become less empathetic and far more confused about what is truly valuable, not because we have physical brain damage like the patients in the study, but because we are voluntarily feeding the conductor bad sheet music.
It connects right back to Lockwood's finding about effort, too.
How so?
If the context is weak and digital, the VMPFC runs a calculation and says, not worth the physical effort.
It's easier to just scroll past a tragedy online than to actually engage or help, because the VMPFC isn't getting the multisensory signal that this tragedy is actually real.
The value computation fails because the context is impoverished.
Something to seriously mull over the next time you find yourself deciding whether to leave a snarky, thoughtless comment online or just keep scrolling.
Indeed. We need to consciously keep our signal-to-noise ratio high in a very noisy world.
That is all the time we have for this deep dive into the ventromedial prefrontal cortex. A huge thank you to the researchers behind these incredible foundational papers we discussed today.
The science of connectomics is moving so incredibly fast right now. It is a thrilling time to be studying the brain.
Until next time, keep your context clear.
And your values aligned.
Take care, everyone.